Today we're kicking off a deep dive that, well, it really started with a pretty startling image. Picture this. An 8-year-old boy, George Huntington. He's riding in a horse-drawn carriage,
Hmm.
and he suddenly sees two women, a mother and daughter, moving in this really unsettling way, almost like an uncontrollable dance. He described it as bowing, twisting, grimacing.
Wow.
What could possibly cause something like that, this shared affliction, that really powerful image sets the stage for our deep dive today into Huntington disease.
Yeah, it's quite a starting point, isn't it? And it really highlights why this topic is so important. We're focusing here on neurogenetics, basically, how our genes affect the nervous system,
Right.
and specifically Huntington disease, or HD, because it's become this sort of cornerstone, a model for understanding these really devastating conditions.
Exactly. So our mission today is to trace that whole journey, from that first clinical observation through the pioneering hunt to actually find the gene involved.
Which was a huge undertaking.
Huge. And then into the sort of the intricate molecular chaos it causes inside cells. And finally, looking at the really exciting, almost revolutionary therapeutic approaches that are being developed right now.
And you'll get why HD is so unique for genetic studies. It's what we call 100% penetrant and monogenic.
Which sounds technical, but what it means is if you inherit that one specific gene mutation, you will get the disease, no question.
It makes it a powerful, though obviously tragic, subject for researchers trying to understand genetics.
Right. So by the end of this, you should have a really clear grasp of the key genetic ideas, the experiments that move things forward, the terminology, and how it all connects to real-world treatments and research.
We'll try to connect that theory to the clinic.
Absolutely. Okay, with that roadmap set, let's really dive into the specifics, starting with what exactly is Huntington disease?
Okay, so Huntington disease is what's called an autosomal dominant disorder,
Dominant meaning.
meaning you only need to inherit one copy of the altered gene from just one parent to develop the condition.
Got it.
And its hallmarks are quite distinct. They usually start showing up around age 45, give or take.
Middle age, typically.
Yeah. And from there, it progresses. You see severe behavioral changes, those uncontrolled sort of dance-like movements, we call that chorea.
Which comes from the Greek for dance, right?
Exactly. Plus, there's significant cognitive decline, psychiatric problems, and tragically, death usually follows within about 10 to 15 years after the symptoms first appear.
It's brutal.
It is. And genetically, it was fascinating because it was one of the very first examples discovered in humans of complete dominance. That one mutated copy gives you the full disease picture.
And what's just incredible is that George Huntington himself, way back in 1872, gave this incredibly accurate description.
Long before we understood genes.
Right. Decades before Mendel's work was even widely known, he noted it was hereditary. He saw it running in families, generation after generation. He also nailed the adult onset and, sadly, that it was incurable at the time. That kind of clinical observation is just amazing.
It really is. And those observations sort of laid the groundwork, decades later, for this revolutionary approach in genetics called reverse genetics.
Okay, reverse genetics. What's that about?
Well, think about it. In the early 1980s, scientists were facing this huge challenge. They wanted to find the genes responsible for diseases, but they often had no idea what the gene did or what protein it made.
So how do you even start looking?
Exactly. The leap was to use variations in the DNA sequence itself as markers. These variations were called restriction fragment length polymorphisms, or RFLPs.
Maybe a mouthful.
Okay, still sounds pretty technical. How could random differences in DNA fragments possibly lead them to the Huntington's gene? What was the thinking there?
Yeah, it's clever. Think of RFLPs as like unique fingerprints in our DNA code. Everyone's DNA is slightly different, right?
Right.
So imagine you use these special enzymes like molecular scissors called restriction enzymes to cut DNA at specific sequences.
If there's a tiny difference in someone's DNA sequence where a scissor normally cuts, maybe won't cut there or it cuts somewhere else nearby, this creates DNA fragments of different lengths. Scientists could see these different length patterns using a technique called southern blotting. The brilliant idea was, if one particular pattern, one RFLP, was always inherited along with the disease in affected families,
Then that pattern must be physically close to the actual disease gene on the chromosome, even if you don't know what the gene is.
precisely. It's like finding clues near the scene of the crime. It's pure genetic detective work, linkage analysis.
So RFLPs weren't just markers, they were the key to finding genes based purely on their location.
Positional cloning, right.
Yeah.
How do they apply this to Huntington's?
This is where the Venezuelan connection becomes absolutely crucial. There was the Hereditary Disease Foundation, started by the family of Lenore Wexler, who sadly died from HD.
Her daughter, Nancy Wexler, a clinical psychologist, heard about this RFLP mapping idea and immediately thought of this huge extended family group in Venezuela near Lake Maracaibo with an incredibly high incidence of HD, one of the world's largest pedigrees.
Wow.
She organized trips, built trust, collected detailed family histories, and crucially, thousands of blood samples for DNA analysis. Her dedication was just immense, driven by her own family's experience.
And those samples were like gold dust for the researchers.
Absolutely. Because in parallel, a researcher named James Gusella was busy developing these RFLP markers specifically to try and map the HD gene.
Okay.
One of his markers, called G8, could identify 4 different patterns or haplotypes, which he labeled A, B, C, and D.
So like 4 different versions of that DNA fingerprint.
Exactly. And using Nancy Wexler's Venezuelan samples and pedigrees, Gusella's team hit the jackpot. They found that one specific G8 pattern, haplotype C, was consistently passed down with Huntington's disease in all the cases they studied from that large family.
That's the linkage, the aha moment.
That was it, a really strong linkage. So the next step was obvious. Figure out which human chromosome carries this G8 marker, because that would tell them which chromosome the HD gene was on.
How'd they do that?
They used a technique involving something called somatic cell hybrids. These are lab-created cells that are a mix of human and mouse cells.
Huh. Okay.
Each hybrid cell line keeps a different, unique combination of human chromosomes. By testing these hybrid cells, they looked for which ones contained the G8 marker fragments. And they found the G8 fragments were always there when human chromosome 4 was present in the hybrid cell, and never there when chromosome 4 was missing.
Chromosome 4, that's incredible.
Monumental. It was the very first time an RFLP marker had been used to map an autosomal disease gene, a gene not on the sex chromosomes, to a specific chromosome. It basically launched the whole field of positional cloning.
But finding the chromosome wasn't the end of the story, right? It still took a while to find the actual gene.
Oh yeah, a long while. Despite knowing it was on chromosome 4, pinpointing the exact gene turned out to be incredibly difficult. It led to this unprecedented international collaboration, the Huntington's Disease Collaborative Research Group, or HDCRG.
Right, I read about that. Huge group, like 58 scientists.
Yeah, across multiple labs, 2 continents,
Yeah.
just a massive effort. And even with all that brain power and resources, it took another 10 years to finally isolate and clone the gene.
10 years. Just shows you how complex mapping the genome was back then. But they got there.
They did. They painstakingly narrowed down the region on chromosome 4 and finally identified a gene they called IT15. We now know it is the HTT gene.
And the key discovery within that gene was?
The repeats. They found that in unaffected individuals, there's a section within the HTT gene with a sequence of 3 DNA bases, CAG, repeated over and over, usually between 11 and 34 times.
Okay, normal range.
But in people with Huntington's disease, that CAG repeat section was dramatically expanded. They had anywhere from 42 to over 66 copies, sometimes many more.
So that expansion, that specific difference in the number of CAG repeats, that was the mutation causing the disease. Finally pinpointed.
Exactly. What a breakthrough.
Yeah.
It directly linked this specific type of genetic variation to the disease itself.
Okay, so they found the gene, they found the mutation, this expanded CAG repeat. Now the question becomes, how does that cause all the devastating problems? Let's get into the molecular and cellular chaos.
Right. So the HTT gene provides the instructions for making a protein called Huntington. It's actually a really large protein, and in healthy cells, it does a lot of important jobs.
Like what?
It's found all over the body, but especially in the brain. It seems to be involved in lots of things like transport within the cell, moving stuff between the nucleus and the cytoplasm, and it also plays a role in communication between nerve cells at the synapses, kind of a jack-of-all-trades protein.
But in HD, the mutation isn't about losing that function, is it? It's something different.
Correct. It's what we call a gain-of-function mutation. The altered protein, which we call mutant Huntington, or mHTT, doesn't just stop doing its job. It gains a new harmful activity.
And that comes from the expanded CAG repeats.
Yes. Remember, CAG codes for the amino acid glutamine. So having all those extra CAG repeats in the gene means the resulting mHTT protein has this abnormally long tail of glutamine residues. We call it a polyglutamine or polyQ expansion.
Polyglutamine, okay. And I think you mentioned HD is one of several diseases caused by these kinds of repeats.
That's right. There are about 14 known trinucleotide repeat disorders where a 3-base sequence gets repeated too many times, and 9 of those, including Huntington's, are specifically polyglutamine disorders.
So what does this long glutamine tail do that's so bad?
Well, here's where it gets really destructive at the cellular level.
Yeah.
That extended polyQ region makes the mHTT protein prone to misfolding. It doesn't adopt its proper shape.
And misfolded proteins are bad news for cells.
Very bad news. And it doesn't stop there. These misfolded mHTT proteins tend to stick together, clumping up into these insoluble blobs called protein aggregates. You can actually see them under a microscope in affected brain cells.
Like little bits of gunk gumming up the works.
Kind of, but it's more insidious than just being physical obstructions. These aggregates seem to trap other important proteins, disrupting multiple essential cellular processes simultaneously. It's not just one thing going wrong, it's a cascade.
A cascade. Okay, so let's break that down. What key things get disrupted? You mentioned specific brain regions are hit hard, like the striatum.
Yes, particularly the striatum and caudate nucleus in the basal ganglia areas, crucial for movement control and other functions. And the mHTT causes problems in at least 4 major ways. First, there's transcriptional disruption.
Meaning problems with reading the DNA instructions.
Exactly. The mHTT protein can actually promote the formation of heterochromatin, tightly packed DNA, which essentially shuts down genes in that area, making them inaccessible.
Locking away the blueprints?
Right. Plus, fragments of mHTT can directly interfere with the machinery, the transcription factors needed to turn genes on. So overall gene expression gets reduced.
Okay, that's one major hit. What's next?
Second, impaired protein folding and degradation. Cells have systems to deal with misfolded proteins. Chaperone proteins try to refold them correctly.
Like quality control.
Yeah. But mHTT seems to mess with the chaperones, making them less effective. And then there's the cell's recycling system, the proteasome, which normally tags and breaks down damaged proteins.
Right.
Those mHTT aggregates can actually clog up or inhibit the proteasome. So misfolded proteins, including mHTT itself, just accumulate, which is toxic, and eventually triggers cell death pathways.
So the cleanup crew gets overwhelmed and blocked. Number 3.
Third is synaptic dysfunction.
This is really interesting because subtle motor problems can actually show up years, even decades, before widespread neuron death.
Why is that? It seems mHTT interferes directly with communication between neuron synaptic transmission. Studies in fruit fly models of HD showed problems with the packaging and release of neurotransmitters, the chemical messengers.
So even before cells die, their ability to talk to each other is impaired.
Exactly, leading to those early subtle symptoms. And finally, number 4, impaired mitochondrial function.
The cell's power plants.
Right. mHTT can bind to the outer surface of mitochondria, messing up their ability to produce energy, ATP. It also increases oxidative stress damage from reactive molecules and prevents mitochondria from moving to where they're needed most, like the synapses, which demand a lot of energy.
So the cells are basically starved of energy and choked with toxic protein.
It's a devastating combination.
Yeah.
And ultimately leads to the death of those vulnerable neurons.
So we know it breaks down, but you mentioned earlier the exact order of events is still a bit of a puzzle. Like, which domino falls first?
Precisely. Is it the transcriptional problems, the protein handling, the energy crisis, or is it always a combination hitting at once? That's still a key question for researchers. But understanding these disrupted pathways have been absolutely crucial for developing potential therapies.
Which brings us to the more hopeful part. The cutting-edge treatments being explored, and animal models have been key here, haven't they?
Absolutely essential. You just can't do these kinds of experiments in humans. So models, from yeast and flies all the way up to mice and now even larger animals, let researchers study the disease progression, isolate specific symptoms, and, crucially, test potential treatments in a controlled way.
Mice seem to have been the workhorse for a long time.
They have. Early mouse models engineered to carry the human mutant HTT gene were vital. They developed motor problems, brain changes, and, importantly, those characteristic protein aggregates we see in humans.
And could researchers manipulate the gene in mice?
Yes, they could create mice with different lengths of the CAG repeat. And they found a clear link. The longer the repeat, the faster and more severe the disease onset and neuronal loss, just like in humans. It validated the model.
Was there a particular mouse study that really stood out?
Oh, definitely. A really groundbreaking study was done by A. Yamamoto and colleagues. They created what's called an inducible mouse model.
Inducible meaning?
Meaning they could turn the mutant HTT gene on or off whenever they wanted, usually by giving the mouse a specific drug, like an antibiotic.
Okay, so they could control the cause.
Exactly. And here's the amazing part. They let the mice develop HD symptoms, motor problems, aggregates forming in the brain. Then they switched off the mutant gene.
And what happened?
The protein aggregates actually cleared up. They disappeared from the brain cells. And even more incredibly, the mice's motor symptoms reversed. They got better.
Wow. That's huge. Reversing symptoms after they've started.
It was the first real evidence that maybe, just maybe, if you could stop the production of the toxic protein early enough, the brain might have some capacity to recover. It suggested that treatment might not just slow the disease, but potentially reverse some aspects.
Now, that mouse result is incredibly hopeful, but how likely is that kind of reversal in the much more complex human brain, especially after years of damage? Are there key differences?
That's the critical question, isn't it? The mouse brain is simpler, and the disease develops much faster. Human neurodegeneration happens over decades. So a complete reversal, like in the mice, might be less likely, especially in later stages.
Right.
But it strongly suggests that the damage isn't necessarily permanent, especially early on. It shows the brain can clear these aggregates and regain some function if the toxic insult is removed. It provides a very powerful rationale for therapies aimed at lowering mHTT levels, particularly if started early.
Okay, that makes sense. It's proof of concept.
Yeah.
And are there approaches beyond just stopping the damage, maybe trying to repair or regenerate?
Yes, there's work on that too. For example, using stem cells. Human mesenchymal stem cells, MSCs, can be genetically engineered to produce extra amounts of helpful factors, like brain-derived neurotrophic factor, or BDNF.
BDNF, that helps neurons survive.
Exactly. When these engineered MSCs were injected into the brains of HD mice, they saw an increase in new nerve cell growth, the mice lived longer, and showed fewer HD-like behaviors. So regenerative approaches are also being explored.
But mice aren't perfect models. You mentioned limitations.
Right. Their brains are small, lifespan's short, and the pattern of neurodegeneration isn't always identical to humans. That's led researchers to develop models in larger animals, like sheep and mini pigs.
Why larger animals?
Their brains are much closer in size and complexity to human brains. They live longer, allowing for longer-term studies. And importantly, you can use the same brain imaging techniques, like MRI and PE scans, that are used in human patients.
So you can track disease progression and treatment effects much more relevantly.
Exactly. These larger animal models are starting to show patterns of brain changes and movement disorders that look much more like human HD, making them really promising for testing drugs before they go into human trials.
Okay, so with these models helping, the main therapeutic goal seems to be tackling the root cause, that mutant Huntington protein. How are scientists trying to do that? Gene silencing sounds futuristic.
It does, but it's becoming reality. The idea is to specifically reduce the amount of the harmful mHTT protein being made while leaving the normal, healthy Huntington protein alone, if possible.
How do you target just the bad one?
Several ways. One approach uses tools called zinc finger nucleases, or ZFNs. These are basically engineered proteins designed to bind to a specific DNA sequence near the mutant HTT gene and block it from being read or represses transcription.
Like putting a roadblock on just the mutant gene's starting line.
Pretty much. Early studies in mouse cells and even cells from HD patients showed ZFNs could significantly reduce mHTT levels without messing with the normal copy or other genes. Injecting them into HD mice also lowered mHTT levels without causing obvious side effects. So, a good proof of principle.
Okay, ZFNs. What else?
Then there are antisense oligonucleotides, or ASOs. These are really exciting. They're short, synthetic strands of DNA designed to stick very specifically to the messenger RNA, the mRNA blueprint, that's made from the mutant HTT gene.
So they intercept the message before it becomes protein.
Exactly. When the ASO binds to the mHTT mRNA, it creates this sort of hybrid molecule that the cell recognizes as abnormal. An enzyme called RNase H comes along and destroys the mHTT mRNA.
Gone. No protein made from that message.
Right. And the clever part is the ASO is then released and can go find another mHTT mRNA molecule. So a small amount of ASO can trigger the destruction of many mutant messages.
That sounds incredibly precise. Has it worked in models?
Dramatically so. Studies where ASOs were infused directly into the cerebrospinal fluid, the fluid surrounding the brain and spinal cord of HD mice, showed a significant drop in mHTT protein levels for weeks.
And did it help the mice?
Yes. Even months later, after the mHTT levels had started to creep back up, the mice that got the ASO treatment still showed improved motor coordination and behavior compared to untreated mice. Similar positive results were seen in monkeys, too.
That's really promising. Are these in human trials?
They are. The first phase A human trials, using ASOs delivered into the cerebrospinal fluid, started back in 2015. It represents one of the first potential treatments to directly target the underlying cause of HD. Huge hope riding on those.
Absolutely. And then there's the ultimate approach, maybe actually editing the gene itself.
Right, genome editing. This is where tools like CRISPR-Cas9 come in.
Yeah.
It's probably the most talked about gene editing system right now.
CRISPR, how does that work for HD?
So CRISPR uses a guide molecule, a small piece of RNA, to direct an enzyme, usually Cas9, to a very specific spot in the DNA. Cas9 then acts like molecular scissors, making a precise cut.
And cutting the gene can? But inactivate it?
It can. The cell's repair mechanisms might fix the cut imperfectly, essentially scrambling the gene's code and inactivating it. Or you can provide a template to insert or delete specific sequences.
For HD being dominant, you'd want to target just the mutant copy, right? Leave the healthy one alone.
Ideally, yes. That's the challenge and the goal. Some researchers, like Jongmin Lee's group, showed they could use CRISPR to cut out a huge chunk, like 44,000 DNA bases, specifically from the mutant mHTT allele in human cells, effectively silencing it while leaving the normal allele untouched.
Wow, like surgically removing the bad part of the gene?
Pretty much. And another team, led by Xiaojiang Li, used CRISPR in live HD mice to actually edit out the expanded CAG repeat section from the mutant gene.
They corrected the mutation in living animals.
Effectively, yes. And the results were striking. The mice showed significant improvements in motor skills, balance, coordination, movement, and this correlated with the clearance of those mHTT protein clumps in their brains.
That sounds almost like a cure, at least in mice.
It's incredibly exciting potential. Genome editing offers the prospect of a one-time permanent fix, but, and this is a big but, we're still early days for using it therapeutically in humans.
Safety concerns, off-target effects.
Exactly. We need to be absolutely sure that CRISPR only cuts where we intend it to and doesn't cause unintended mutations elsewhere in the genome. So while the potential is immense, more research and rigorous safety studies are definitely needed before we see CRISPR used to treat HD in people.
Okay. So as we wrap up, it's really quite something to trace this whole story. You know, from George Huntington seeing those women moving strangely, all the way through figuring out RFLPs, finding the gene on chromosome 4, understanding the CAG repeats, the protein aggregates, the cellular chaos.
Decades of painstaking work.
And now, these incredibly sophisticated therapies, ASOs, potentially CRISPR targeting the very root cause. It's just an amazing scientific journey.
It truly is. And the way the researchers tackled HD, combining family studies, molecular markers, animal models, cutting-edge genetics, that whole integrated strategy has really become a blueprint, a paradigm for how we approach other complex brain disorders, too.
Yeah, the lessons learned from HD research extend far beyond just this one disease.
Definitely.
But even with all this incredible progress, there's still that lingering question, that thing to maybe mull over. We know all the things that go wrong inside the cell because of mutant Huntington, the transcription, the protein folding, the energy, the synapses. But what's the trigger? Is there one key disruption that kicks off the whole deadly cascade? Or is it always that complex, messy combination of everything going wrong together that ultimately kills the neuron?
That's still the puzzle, isn't it? Understanding that precise sequence, that initial trigger point, could be key to developing even more effective, perhaps even preventative therapies down the line. It's what researchers are still actively digging into.
A fascinating question to leave things on. Well, thank you for joining us on this deep dive into Huntington disease. It's been a journey through some challenging but also incredibly hopeful science.
It really has. Thanks for exploring it with us.
We hope you found this look into the genetics and potential future of HD treatment insightful. Thanks for being part of our deep dive today.